We get experimental science. We're curious, non-judgmental.
Let us bring in our very special guest today. Dr. Elizabeth from the Fred Hutch Cancer Center is going to be on live with us to talk all things blood, cancer, and more. Bring her on the stream. Here she is, chat. There is our special guest today, Dr. Elizabeth from the Fred Hutch. Elizabeth, how are you doing today?
I am well, and unfortunately I'm going to have to correct you. I am soon to be doctor and not quite yet doctor.
Soon.
But thank you for boosting me.
Well, listen.
Early.
Soon to be is you're almost already there, right?
Just a stone's throw.
Yeah, exactly. You know, you're already broken yourself enough to the point where we can refer to you as being fancy. But thank you so much for coming on with us today to talk science about your research, as well as Elizabeth sent us samples that we're going to be looking at under the microscope today. Blood samples, cancerous and non-cancerous cells, I believe. So
you got that right. So they're bone marrow samples, so the source of blood from some of our model organisms.
Yeah, so we're going to be looking at them in a scope, y'all. We tested out the samples, the scope. It looks great. We're going to be getting y'all to see some really, really nice, detailed science today. Just a couple of programming notes before we get started. We had 2 donations already from, again, Tactics and Travel the World. Thank y'all very much for those donations. We do have some stretch goals set up. So last week we hit our 2-stream goal of $1,000 in 1 stream. So we added in a couple of stretch goals. The stretch goals are if we make 1,500, we will do a couple of extra raffles today. So last week y'all really enjoyed these coffee beakers. All the hype for the coffee beakers. So we'll be doing, we have 2 coffee beakers to raffle off today. One is for every $25 donation today. You'll get entered for a chance to win a coffee beaker that we'll pull at the end of stream. If we hit 1,500, we'll do another raffle for a coffee beaker that is independent of any donation amount. So you do not have to have donated anything to get in for that $1,500 mark. We also have, folks, 2 t-shirts on the line. One of these beautiful Fred Hutch Cancer Center shirts that I'm sporting today. Again, one of them will be for every $25 donated, you get entered. If we hit 1,500, we'll have another independent raffle of that. And finally, if we hit the ultra super stretch goal of $2,000, we will do a raffle for a 30 million year old amber piece with an insect in it. So we're putting that up on the line, folks, to be able to do that too. So that is what's on the docket today. And we've already been kicked off in the starting student screen by Travel the World and Tactics. Thank y'all very much for your donations. And hello, everybody in chat today. I'm sorry I've been missing some of the chat, folks, but welcome the heck in. We had Zedman here first today. We've got our friends from Fred Hutch here, Daka, Music Girl. We've got Smikes24. We have ADC Art Attack. Luminology is here. Adrian No Art is here. Astro Canuck. We've got all the party people. Coffee Rocks, Jaded Fairy, BitGamey. Oh my goodness. Some Mike Somewhere, Dreaming Truth, Clayman Dev. A lot of our friendos are here. Thank you all so very much for coming on and supporting science, as well as the charity stream we've got. And the dono link for anyone interested is pinned to the top of the chat, or you can type !charity, and that will also bring up the link for the Tiltify. And hello, Skip the Pickle as well. Welcome in. If you didn't know, Elizabeth, our friend Skip the Pickle is actually a pickle VTuber. Who does not like pickles. Does not like pickles.
What?
I know, yeah. It's he means skip the pickle, as in do not eat the pickle.
Oh, well, that doesn't sound very tasty at all.
I know, right? It's very surprising to me. But Elizabeth, thank you again so much for coming on. Please tell us, give us a little introduction about who you are, how you got into science, and a little bit about what you work on.
For sure. My name's Elizabeth. I am a graduate student here at Fred Hutch. I study through UW. And how I got into science is actually a little weird. I'm old enough that charter schools weren't a thing when I went to high school, and I actually didn't have science or math in my high school. I went to one of the first charter schools back in Denver, Colorado, and high school didn't agree with me, and I wound up leaving and just bartending for years and years. And I was working one Christmas, and a customer shot my car up with a gun, and I was like, well, I don't want to die in a bar, so I'll go back to school for nursing. And I took my very first science course at I think 25, and I was sitting in the classroom, and I was just like, wait a minute, like this is so freaking cool. Like, how did I not know about this? And I fell in love with science, and so that's how I find myself here now working on my PhD at Fred Hutch, is just kind of through random happenstance.
Can I ask what was that class that like what kind of science class was it that got you excited about it?
It was a like the it was like biology 111, so like your introduction to cell biology. And I think it was like protein folding, or it was something weird, or I really hate to admit this, but maybe something to do with like metabolism. Like it was one of just those weird cellular processes when I really realized kind of what goes on inside a cell, and like how these very small pieces that kind of make up the whole of all living things were like just insanely complicated. Like they're like their own like little worlds, and kind of this idea of like there being this microcosm of existence, and that kind of builds us all up. And I don't know, this might also be a dated reference, but I think back to like, I think it was like Men in Black, where there was like universe inside of universe inside of universe, and like that kind of like hit me, and it was just done. And I've been here ever since. So
I love that story. That is so cool because usually like we've heard of people getting into science, but it's like the neuroscience class or it's like these like very specialized course, and it's never like that intro bio class. It's usually like where people are like, no, I didn't like bio after that. But it's super cool to hear like that's what like caught your imagination, is that cell biology to start with.
it's crazy stuff. I think it makes me appreciate how complex life is, even when it doesn't seem so complex. Like it's really an amazing feat that any of us exist at all. So
Well
I
said.
think that's what cell biology has kind of taught me.
And actually, while we've been chatting, we've already gotten a couple of donations. We're up to over $1,200. We have $25 from Music Girl, $5 from Legion, $25 from Smikes, and $25 from Dacum. So we are well on our way to hitting one of our milestones. Thank you all so very much for those donations. So, Elizabeth, what got you to go to grad school then? What was the. Because that's another big jump, right? If you're going into
It-
sciences.
yeah. Well, so I mean, I'm sure part of it was that like it's like you finish your undergrad degree, and then I wound up working in a lab. So I came from Colorado. I worked in a lab back at Anschutz Medical Campus, part of the University of Colorado family. And I really, really loved the work I was doing. I was working in solid tumors. And at the time that I started working in this lab, my godmother got very sick. She was diagnosed with cancer, and we all thought she was going to be okay. We thought she was going to be okay, and then in a matter of a year, her tumor was just exceptionally aggressive. She was gone. And so kind of that experience of like, even she was getting treatment out in New York at Memorial Sloan Kettering, which is like got this reputation for being like one of the best treatment centers in the world. So even with the best doctors, there wasn't a lot of options for her, and she was doing clinical trials. And so for me, and there's so many of us who have been touched, I'm sure, by cancer. So for me, a big part of it was, well, how do I improve patient experience, and can I help make a dent? Because it's brutal. It's really hard to watch somebody you care about get treated with drugs that, while they're saving your life, just also make it really miserable. So for me, grad school is an opportunity to kind of take that next step, take kind of this passion that I have and then use that to hopefully develop things that make our lives better. So
That's an
that's how I
amazing
found this
story.
idea.
Thank you for sharing that, because I think it also highlights that kind of there's a disconnect with researchers sometimes, right? It feels like we're almost not necessarily human like everyone else, that we're just like in a lab and just pipetting away, and it's like what everyone's motivation is for working on a particular question. I think that's huge, that that's what like motivated you and to got you excited, is to even like because of a family member. And so that's really really incredible. So you were working in solid tumors, and now what are you working on? What did you get to?
Yeah, so I went from solid tumors to some really hardcore cell biology where I was just studying this one molecule, and I really loved the tumors, which is a weird thing to say. I really loved kind of this application of science for the betterment of people, but then I also found this one molecule absolutely amazing, and like I fell in love with this one molecule. And I came to grad school, and they were like, try a bunch of stuff out that you didn't think that you would like, and you'll find something. So I did that, and it just didn't work. But I managed to find this really amazing mentor, and this lab that was kind of taking this molecule that I think is just the coolest molecule on the face of the planet, but then also trying to study that molecule in the context of cancer, and really work towards how can we use this or leverage this to really build therapeutics for people who have very specific types of cancer. And in my lab currently, we study blood cancers, specifically blood cancers that generally tend to affect older individuals. And as I get older, that also feels very applicable because it comes on fast.
Can you tell us a little bit about what blood cancer is? Like that there are differences, like you said, between young and old. Like just a little bit of the background, and is it like already in chat where one folks are wondering, is that like a form of leukemia? Like so, like when you say blood cancer, like what does that manifest as, and what are some of the differences between like young and old and the different types?
For sure. So leukemias absolutely are a type of blood cancer, and there are definitely different types of leukemias. So what blood cancer basically is that you have these cells that are in your bone marrow that kind of give rise to all of these cells that are within your peripheral blood. So these are the cells that are responsible for fighting infections and for carrying oxygen throughout the body, and then also the cells that help you clot, so help you stop bleeding. And basically, what any blood cancer really is that these cells within the bone marrow, at some point along this passage from being like this very basic cell, this stem cell, to being kind of, a B cell, or, an oxygen carrying cell, a red blood cell, something goes wrong. And they don't turn into those cells, they get stuck kind of mid path, and they just grow out of control. And they can take over your bones. They can take over other organs that hold those cells, and basically they can make you really, really, really sick. So leukemia, I think, is the blood cancer that everybody is the most familiar with, and I think that's predominantly because of things like childhood leukemias, which are absolutely horrendous, and are really detrimental to young people. But as we age, there's this huge group of cancers that kind of occur, and there are two different types. One are kind of the leukemias, and then kind of the precursor to that and tangential to that, it's the blood cancer that I study, which are known as myelodysplastic syndromes, or MDS, which we'll just call it so nobody has to remember myelodysplastic syndrome. And basically, MDS is where it's not necessarily as severe as leukemia, but you kind of lose all like these really important peripheral blood cells, and you have this accumulation of these cells in your bone marrow, and it basically, because you're older, it really limits your longevity because you've stopped producing oxygen carrying cells, or you're no longer clotting appropriately. And when we think of our older relatives, and we're already worried about things like slip and falls. If you're not clotting appropriately and you have a slip and fall and all of a sudden you're bleeding more, or you're more prone to slip and falls, or, you're more prone to infections, all of these things can dramatically decrease kind of the lifespan as we age. So that's kind of my niche. But blood cancers as a whole, we can just think of like when kind of young cells go bad and run riot in our bones.
That's terrifying, especially like how you're saying with like the kind you're studying too, because like you said, there's already concern about slipping and falling. There's infections already a concern when you're older, so it complicates a difficult part of life even further.
Absolutely. Yeah, and I think we all want to live happy in our old age. I mean, at least I do.
Yeah, and Smikes in the chat says it almost sounds like a comorbidity question, almost like having even before this blood cancer might go metastasize, right? It seems like it could just very easily cushion you along towards a bad end.
Absolutely. Absolutely. Yeah, there's different levels to how bad it is, but there's absolutely a shortening in life. And then again, I think quality of life is really important. I have a parent who is approaching 80, and if there's one thing we want for both people that we love and ourselves as we get older is like, I worked hard in my youth. Like, I want to be happy and comfortable when I age. I don't want to have to be going in for blood transfusions every other week just to make sure that like all of my limbs have oxygen, or if I cut myself making my favorite meal, that like it will stop. So
Is that the current
it's
treatment option? Is transfusions?
yeah. So for MDS, there's kind of like you can do kind of like these things to kind of stave stuff off, where like you're going in for like regular blood transfusions. The only actual curative treatment is what's known as a bone marrow transplant, which was actually invented here at Fred Hutch. This is also one of the only curative treatments for leukemia, where basically you use like high doses of these really toxic agents to like eliminate all of the cells in your bone marrow. And then you find like a somebody who's got similar bone marrow to yours, and you get it donated, um, which is really not possible for a lot of people. One, because finding someone who's willing to donate their bone marrow, which is an exceptionally painful process. They take this huge needle and then jam it into your bones, and like it just sounds horrendous. But you have that limitation of, can I find somebody who's willing to get a needle jabbed into their bone? Or the other issue is age. Like, is the patient healthy enough to survive this potential treatment option? So it's not super great.
Yeah, that. Yeah, the prognosis does not seem good. So, we have members of chat also are mentioning that they also had loved ones lost to blood cancers, and of how it's not a rare disease, right? It does come pretty often, and so
Absolutely.
yeah, it's super important
Yeah.
work that you're doing. And last week we actually were talking about how much like a donation actually will help Fred Hutch, and something like $5 helps get 100 test tubes. It was like actually putting that numerical amount on how much lab equipment can help, and I think it was like $1,000, or no, was it? I think it was maybe a 500 that allowed for like a sequencing experiment to be run as well. So it was like putting those numbers on like how much you'd be able to get out of is pretty remarkable. So every donation counts, y'all. So actually we've gotten some more. We got another 25 dono from Coffee Rocks in the chat, and another 10 from Tactics. Tactics was very much like in on the beaker raffle, as it were. But another question from Dacum: does blood marrow replenish itself in a healthy person? So if one were to donate, you're okay if you donate?
Yes, absolutely. So the brilliant thing about these blood producing cells, these blood stem cells, is you can imagine that when you're an infant, you've got small bones and a small body, and you need less blood and you need less kind of everything. And then as you age, you have greater space and you have more need. You have, more need for blood. You have more need to fight off infections. So these blood stem cells are not only capable of producing cells of mature lineages of these, peripheral blood cells, but they're also able to replenish themselves. So they're a really cool population of cells. Stem cells from kind of anywhere in the body are really quite amazing because they can kind of do these two different things where they repopulate and they also make other cells, which other cell populations can't necessarily do. They're either themselves and kind of stay themselves and kind of reproduce, or they replenish. So they're really interesting population of cells to study.
And because they're stem cells, right, it's all the more likelihood that when something goes wrong, it keeps getting worse and worse, right? Because they're self-renewing. So if you have some mutation early on in that stem cell stage, you're just going to proliferate the bad kind of blood cell throughout the body.
Yeah, absolutely. And that's the hypothesis. That's kind of what we think is happening with these age-related cancers, is actually that in these blood stem cells, probably sometime between 40 and 50, you might be picking up these mutations and these errors in your blood stem cells. And what happens is that over time, they kind of accumulate and accumulate and accumulate, and then all of a sudden you see the effects that they're fairly slow, unlike a childhood leukemia where your blood stem cell goes bad and it's just like it's anarchy immediately. So yeah, the any error in a stem cell is it's a bad place to have an error.
Yeah, so this seems like a very scary population of cells to have that error in. Another question.
Yeah. Well,
oh, go ahead.
yeah. Oh, I was actually going to say it's there's kind of a theory in the field of cancer that all cancer actually takes cells and turns them into a kind of a stem-like state, even though they're kind of maybe more mature cells.
So it's just acting along what it would be naturally doing, right, in terms of the cancer spread. The question from the chat is, what is the Hutch doing to change the treatment process of blood cancers, which is pretty much poisoning the marrow? And I think you can comment on that in some of your research. Should we shift gears and pull up some slides and take a look? All right, chat. So we're gonna jumble around now. I have to jump over to the other side, so we're gonna move Elizabeth over on my side and vice versa. So Elizabeth is gonna come here. I'm gonna hide. Look at that. Easy, no problem. I promised her it would be very painless. Although, Chad, she did mention she watched some of our VODs and that we're a little bit chaotic here. So y'all have been very behaving today. Thank y'all.
Thank you.
There we go. We've got some of the blood cells that you sent us. Let me go ahead and crank the lighting up a little bit. So we've actually all got 2 samples today.
Together, we can rule the galaxy.
Thank you for the subscription. Thank you.
Get good 609 just subscribed with Prime.
Thank you, Gig. A follow and a subscription. Thank you very much. So these are some of the cells that you sent us. What are we looking at, Elizabeth? Like how do you know? Because I know you've been trained at this, right? You look at these all the time. But as I'm used to insect cells, like of ants and flies, like walk me through, what is this stain? What are we looking at? How can you tell what we're looking at? How can you tell if it's healthy or not healthy? Like, tell us all the things.
Absolutely. So these are bone marrow cells from one of our model organisms. So we use several different models to study blood cancers to figure out how to better treat this. So this is from one of our mouse models of leukemia. And what we're looking at are the blood producing cells. So these are either blood stem cells, or these are more immature cells. In blood, we have several lineages. We have this myeloid lineage and this lymphoid lineage. And we won't go down that rabbit hole unless people want. But so mainly what we're seeing are these kind of immature progenitor cells of this myeloid lineage, as well as our bone marrow cells. And basically, we're just seeing a lot of immature cells. The way we know that these are healthy and happy is, everyone is fairly round and fairly even. And I don't know if you can see. Can we zoom in? Yes,
We can,
zoom
yeah.
in a little.
They adjust the lighting for us too. Take off that auto exposure. Go.
There we go. So if we kind of look at these images are comprised of two parts. We've got kind of like the circle, and then like the little like kidney bean looking thing in the middle. So the
this
kind of the whole,
bleb inside.
yeah,
Okay.
the little kidney bean deal. So the big round circle is basically the whole cell, and the little darker purple like kidney bean deal is where cells hold all of their genetic information, called the nucleus, if people aren't familiar with the nucleus. And so basically we can tell these cells are healthy and happy because the cells are all fairly uniform and the nucleus doesn't look super strange. And if you look at the cells, there's one up in the corner over here. Where is it? Right, I think I'm pointing at it. It's a little hard to adjust. That cell with the hole in the center, that's a dividing cell. So that means that cell is going to go from being one cell to two cells. But we're not seeing a lot of that. We don't want the cells in our bones to divide too fast. They should basically be dividing enough to kind of replenish themselves, or to, start making downstream more mature cells. So we can kind of see from all of these cells that everybody's fairly uniform.
Gotcha. That's really cool that you can tell the dividing cells so easily as well. Just like that morphology, like the structural information that we have here. Like, let me see. Here we go. So like you would think like that one right there, where it's like that circle's in the middle of the cell, like that's another dividing one.
Yeah, it's getting ready to pop off and make a daughter cell.
Nice.
And because this is the bone marrow, unfortunately, we don't see a lot of different types of more mature cells. I looked for slides with like all of that stuff, but I couldn't find any that were good enough for this fine audience here. So yeah, we would use this as a control. So we would take our mice, and maybe we would do a genetic perturbation, or we would change something around, such that we would expect that if whatever we're changing were to cause disease, that this morphology would change. And then we would have some mice where we would just kind of let them rock and roll, and then this would be how we would tell, from this group of mice, that these mice are healthy, and that either the treatment we're giving isn't affecting the bone marrow, or that the mice themselves are just sporadically developing leukemia.
Couple of quick questions we've got. So the nucleated cells, everything here is nucleated, right? It's just that we can see some are dividing and some are not. So there's nucleated cells up in the other areas too. They're just not yet dividing.
Yes, that's correct. So you will have enucleated cells, which are cells without a nucleus, and those will be your red blood cells. But basically, when the cells kick their nucleus out and become the cells that carry oxygen, they book it out of the bone marrow to go and do their job. So we don't generally see a lot of those in the bone marrow.
That's like the more mature stage, right? Where it's like you've already differentiated into the cell that you need at the end of the day to go into your bloodstream.
Yeah, that's correct. Once you don't have a nucleus as a cell, there's not really a whole lot you can do anymore. You've lost all your information. So
yeah, so just for the folks coming in, we're talking about blood cancer today with Dr. Elizabeth, soon to be Dr. Elizabeth, soon to be. She corrected me, Chad. We can't jinx it. And we're talking about the structural information right now of wild type normal blood cells within the bone marrow. So these are our stem cell populations, what they end up dividing. And we're going to look at some sick cells as well. We had two more questions. One is, you mentioned that these are with mice. Do you have to use mammalian model systems to study the questions that you're asking? Yes or no? And then what other organisms can you use to study these questions?
These are all great. Those are several great questions. So flies have long been used to study kind of everything. So fruit flies, they're a little further away and don't necessarily produce the same types of cells, but people have used flies. Another really cool model for development that people use are zebrafish. And what makes zebrafish so cool is that when zebrafish are embryos, before they become like fish, they're transparent. And so what people can do is they can label different cell populations or different proteins with different colors. And then as the zebrafish develop, you can actually watch how these cells change and move, and like they're super cool organism. And again, they're a little further away from people, so they're not necessarily as great for modeling human disease, but for studying like the development of the blood system, they are absolutely amazing. And because you can actually visualize in 3D how these cells kind of move about and arise, and how they take hold in the body. It's really pretty cool. So there's a bunch of different organisms. And honestly, the field currently, I think, is trying to move away from using model organisms. So there's some work trying to build like bone marrow on a chip. So you build 3D structures with 3D printing, and like have all sorts of different cell populations in there. And can we like simulate the bone marrow without having to use any animals? And then there's like these things called organoids, where like if you just put all the cells together, will they, and like give them all the right goodies, will they spontaneously form like a bone marrow like environment? So there's a lot of different really cool models being used in the field.
Nice. So it's like getting closer to the actual human side of things versus modeling it as something a little bit more distant.
Absolutely. And I think ultimately none of us want to use animal models. We want to honor all scientists generally want to honor all living things. So it ideally we would study what we want to study, in the most native stage we can, and do it with causing as little harm as possible. So
Right, totally. Um, we had another question from Coffee Rocks is, when we're looking at this kind of slide, what is between the cells? Are we looking at lymph here? Or is it just the cells spread out? Like how did we get this sample, and what exactly is, like in terms of like just orient us of everything else outside of the cells, like what else is on this slide?
so it's basically salt water that's around these cells. So basically what we do is when it comes time and we collect the organs, so when we collect bone marrow, we'll take that and we'll wash it really good, and we'll wash all of kind of the bits and pieces off. And then we resuspend it in basically like a saline, a buffered saline, so that the cells don't rupture. And then we just spin them, like smush them down onto the cells, onto the glass slides, using centrifugal force, and then fix them with some alcohol, and then stain them with two different stains, which kind of allow us to see the different parts of the cells. So kind of around them, there's not really anything else. If you're seeing particulates and bits and pieces, it's just leftover debris. But we can't really visualize the fluid on a slide, so we just try and get rid of all of like the gunk, because otherwise it would be hard to see kind of what we're really looking at.
So we try to just isolate. You're just wanting the cells for these preps, so you can. That's what the phenotype is that you're going to be looking at. What are the
correct.
two stains?
So it's the kind of globally, it's called Giemsa staining. And I honestly, should know what these two stains are. They bind to two different types. I think one binds to like a protein, and then one binds to something else, and they're two different colors, and it's probably based on like acidity. But it's a very old school technique, and unfortunately for some of us newer scientists, we're like, yeah, it just works, just do it, and it's not great. I really should know how this works. Don't tell my boss.
I'm just curious, we're just asking away, and it's totally like I think one thing that folks appreciate too is when people say I don't really know, right? This is just a technique that we do. It's I think important to demonstrate that scientists aren't just like in this ivory tower and we know everything, but versus like there is a lot of unknown. We're just like any other person, and it's a lot of active learning and coming up with what the answers are like as you're working through them. So yeah, for those of y'all coming on in, we were doing another charity stream with Fred Hutch today. We last week raised over $1,000, so we're back for part 2. This is Elizabeth. She works at the Fred Hutch Cancer Center on bone and blood cancers. Well, so I say bone because the bone marrow is associated with it and the blood cancer. She'll be telling us about some of the treatments that she's working on as well, which hopefully will combat some of the poisons that we currently have to use today. We also have the dono link pinned to the top of the chat. If y'all would like to donate, we'd love to have any donation. Will make a huge difference. You donate $25 or more. Every $25 gets you an entry into a raffle at the end of the evening, which will be for a Fred Hutch beaker, as well as a t-shirt. And if we hit that 1,500 mark, we'll do 2 of each of those raffles. If we get to 2K, we'll do a raffle of one of our 30 million year old amber pieces with an insect in it as well as our stretch goal. No pressure at all, folks, but that is what we're doing today. And already, thank you to our donators throughout the stream already today. We had another question, Elizabeth from Smikes, who was wondering, let me scroll on back here, is synthetic marrow something that you think ever could happen?
I'm going to err on the side of optimism and say yes. It will, like anything we make, it will have limitations, but I mean, if I say no, then why am I doing what I'm doing? So the hope is yes, and ultimately, kind of what fuels that is not what I do, but basic research, which I think is harder to get funding for. But the better we understand something, the better we can mimic things, and if we can get 50% of the way there, it's way better than where we're at now, and every improvement is a step in the right direction. So I'm gonna say yes.
Absolutely. And then I don't know if this is outside of your realm of expertise, but Legion was wondering if there's of any dietary benefits of having certain diets that may help combat this.
It's somewhat beyond the scope of to use what was mentioned in the chat. That's beyond the scope of my research. There is some research showing that at least in age-related blood cancers, that it's a highly inflammatory environment. So I've kind of preliminary done some cursory searches on things like diabetes. And MDS, diabetes is an inflammatory disease, and this is an inflammatory disease. So you could kind of imagine that would be a worse prognosis. So there's some indication that, kind of how we treat ourselves long term might have an effect. But in terms of like eat this to not get blood cancer, I think ultimately kind of where we stand in the field is that this is just unfortunately like a case of bad luck. Like you just kind of are unfortunate enough to like pick up an error in these cells, and it there are absolutely other blood cancers and some indications that there may be some inherited mutations, which it's unclear, but we may be able to link those to this, to my certain type of blood cancer. But in terms of diet, I haven't seen any direct links that like, a vegetarian diet will prevent you from getting MDS.
Gotcha. And Ubi, thank you for the $25 donation. We're up. We're past. We're a quarter of the way there for today's stretch goal. Thank you so much, Ubi. I had another question for you. Because this is in older populations, presumably that's easier to get as you age, and if you live long enough, everyone might end up getting this, right? Because the likelihood of getting some kind of mutation in these cell types, again, with time, eventually you're going to get it, right? So this is a very, very important disease to be studying.
I think that at least it's what I write in my grant applications, is that, we have an ever increasing global population. And if it's not, myelodysplastic syndromes, it's an error is going to occur. And as, kind of like with a car, like as the car gets older, pieces just kind of break. So if it's not your oil, then, it's going to be, a drive shaft or a wheel bearing. Or so I think, kind of addressing age related cancers is critical as global population just continues to live longer.
Yeah, so it's
These-
like
these are just going to be-
with the strides that we make in letting people, like having people live longer, has to be paired with, well, now you're more likely to get this horrible disease state that we now have to figure out how to fight as well. So it seems like it's always a double-edged sword, right? Of one benefit, but then a negative might be creeping up on you right there.
Yeah, I mean, the universe is always tending towards entropy, right? Like the universe is always headed towards chaos, and then there's always this kind of counterbalance, right? Like it's creation and destruction simultaneously. So that I think that's kind of the fight we spend our lives with, both both internally and externally.
Absolutely, yeah. So should we pull up the other slides, and then you can tell us a little bit about what you're working on them, or do we need to roadmap a little bit more on what a normal cell looks like?
No, unfortunately, like I said, I couldn't find any peripheral blood slides with, like, so I could show you guys some crazy cells. But yeah, I think we have gone about as far as we can go.
Okay, let's pull up these are. Yeah, let us know what, I guess.
Victory screech!
As we pull up these slides, what is wrong with these cells? Like, what has been done to these?
So these are mice that we've given a mutation to, and basically these mice have developed, they're in a pre-leukemic state. So MDS, the cancer that I work on, can actually transform into a more aggressive cancer called leukemia. And so these mice are right there. So you can see how even in the few cells that we're seeing here, like, let's see, it's hard to orient.
Right. Yeah.
Oh, there we go. So you can see right here this group of 3 cells, 1, 2, 3.
Yeah.
Where that little star just passed. Those were all looking really. So that one's divided. I'll get it. That one's dividing.
Yep.
And then the one next to it looks okay, and then the one next to that has got, like, I don't even know what's going on there.
Yeah.
Like,
So is that the nucleus, the organelles, or just everything is just trashed in here?
That's the nucleus. So
it's like this misshapen.
That's a cell that has decided it's going to do what it wants to do, and it does not
same
care.
like above my head, right? That's that same twisted structure of the nucleus. And so presumably, like, changing the nuclear architecture and how the nucleus is supposed to look is going to have a lot of downstream problems, independent of being this cancer, but just any kind of time where you mess up the nucleus in this state. Like, gene expression is going to be thrown off and not what you'd expect. Like, what are some of the things that are associated with these kind of weird structural issues just in general?
A misshapen nucleus, and specifically there's a structure inside the nucleus called the nucleolus, which is where your cell makes all the things that it needs to, like, work with DNA. So a misshapen nucleolus was actually the first visual diagnostic feature ever discovered for cancer. So
wow.
When so when you see the nucleus, and specifically, we can't see it in these images, but this little structure inside the nucleus called the nucleolus, when that goes, like, wonky, that's how they used to diagnose cancer.
By wonky,
So,
do you mean like is it normally like a circular structure and that gets like twisted, or okay?
yeah, it gets large and it gets like misshapen.
Oh, we have the raid in as well, y'all. Shipwreck Bricks, welcome the heck in to the Raiders coming on here. Shipwreck, how was your stream? Tell us all the things. We are doing a charity event today. With Elizabeth from the Fred Hutch Cancer Center. It may have, yes, our apologies for the alert. That may have been a little much. I did not warn Elizabeth about the sound alerts. Welcome the heck in, y'all. How was the stream? Lightning Builder, hello. Shipwreck, hello. Welcome. What were y'all up to today? The coconuts are migrating from Puck's Peninsula. How are y'all doing today, Shipwreck? Again, we're doing a special charity stream today for Fred Hutch Cancer Center. Guys, go check out our friend Shipwreck Bricks doing some makers and building. Were y'all doing Legos? What were y'all building? I want to know some details. Hocus Pocus. Oh, the Hocus Pocus house. Is that the new set that just dropped from Lego recently? Y'all, and what was your thinking of Hocus Pocus 1 and then 2, which came out recently, Shipwreck? We need to know this. This is Dr. Elizabeth here with us. She works on blood cancer, and it's really cool. We have slides that we're putting underneath the microscope, and she's showing us some of the elements of like what's a cancerous cell, what's a normal cell, and we're getting into the meat and potatoes now of chatting about what she's working on to combat the tumors and the cancer of the blood cells, in particularly older populations. And so, but we get to look at real samples. You can see down below me for the Raiders coming on in, those are misshapen nuclei. So you can see there's this one nucleus that has this giant circle in the center. There's one in the middle. It's like looks kind of okay, and then there's this crazy wonky one, not at all circular, but the nucleus is just totally trashed. So we're raising funds for Fred Hutch Cancer Research Center. A $5 donation gets Elizabeth 100 test tubes that you can do experiments with. $500, there's a sequencing experiment they can do. So every amount that we get goes directly to research. And if anyone donates $25 or more, we're raffling off today. Later in the evening, beakers with Fred Hutch written on them. We also have t-shirts, which I'm sporting one of them. And if we hit our sub goal, that'll be, we'll do a raffle for a 30 million year old piece of amber with an insect in it. And yes, ask chat, Raiders, ask away. Anything that y'all want to ask, Elizabeth is here, graciously giving us her time to be here and chat with us. Yeah. And Risto, thank you for the donation as well, Ristrock. Oh my goodness. Risto coming on in with, I believe, another, let me just double check, Risto with 25, yep, with, $40 donation from Ristrock 88. Thank you so much, Risto, for that dono. How are you doing, sir? Welcome the heck in.
Risto on the shirtless calendar for the orphanages.
He's putting out a shirtless calendar for an orphanage to benefit a nearby orphanage. He's a
Nice.
Philanthropist and a family man. We love Risto here. Guys, check out our friend Risto as well. Coffee Rocks is another question. You said that the cure was the destruction of the bad marrow, followed by a transplant. How long does it take from transplant to function for the patient?
Ooh, I'm not actually too well versed on like what the timelines are. I know that it takes some time because the cells have to move in and kind of take over the bone marrow, and then like once they're settled in, they can start doing stuff. But just because you get the transplant actually doesn't mean that it'll work. So you can develop things like graft versus host disease, where your immune system, what immune system you have left, recognizes like these bone marrow cells that are from someone else and will actually attack your bone marrow and make you really, really, really sick. Or the transplant won't take, like the cells just like decide that they are not happy in your bone marrow. So like that you'll get the transplant, everything will go fine, and it just fails. So I think that it's my guess is that it's individual for patient to patient. However, like I don't know like average times, but I do know that even if you get these transplants, that it won't necessarily work. And they have the potential to make you even more sick.
So do you by chance know like the percentage of success?
I don't.
All right. That's and because you mentioned like the immune system, are you also on immuno like suppressants for the rest of your life? Do you know?
I don't know. I've buried my head in the basic biology probably more
Yeah,
than
it
more
just
than I
seems
should
like
again.
it's already such a hard process of having that bone marrow transplant anyway, and then it's like that likelihood of not working as well is pretty intense of like what can work and what doesn't work, and I guess it makes it frustrating as well for, and all the more important for what your work is to make sure that we make a big difference, right?
Yeah, let's find some drugs.
Yeah. Guppy, welcome the heck in. How you doing, Guppy? Let me see. In an alkaline
Thank you,
environment,
Gabby.
do you know anything about the alkaline environments and how those correspond to cancers?
So I am not sure about pHs. The only thing I know about pH is that your body is very good at maintaining a pH, and you can't really change the pH all that much, or you get very sick. But outside of that, like I think that if you change the pH for any cell type, that it gets very sick. Like even like our cultured cells in like we have these cells that live forever which we've crazy modified, and I know that if we modify pH even for these cells that should withstand everything, they get really sick. So I'm not really all that sure about things like alkaline environments. I know that there's a lot of trending health stuff with that, but I
Gotcha. Well,
je ne sais pas.
let's get to some potential drugs, though, right? So,
Yeah.
what are you trying to do in your research? Because you have these cell types that you're looking at. You have these normal cells. You have these tumorigenic cells. What is it that what is the big question that you are studying?
So for what we're studying, very specifically, is so these cancers arise as a result of specific types of mutations, and what we want to do is these mutations obviously are taking healthy cellular processes, the things that the cells should do really well, and kind of screwing up the works. So our question is, in the process of kind of screwing up the works, can we leverage that as a way of treating sick cells without necessarily harming the healthy cells? And for us specifically, my lab focuses in on one very specific group of mutations, which are actually some of the most prevalent in our specific type of blood cancer, but also occur kind of across a whole host of different solid tumors and blood cancers. And that's mutations in genes encoding the machinery for what's known as RNA splicing. Um, so kind of step back really big picture, kind of this central dogma of biology is that our DNA, which kind of holds all the information for all of the everything, becomes RNA, and then this RNA is what the cell uses to make proteins, and then proteins are the tools that kind of go off and do the work. So I think I'm guessing that a lot of your viewers are kind of familiar with DNA and the idea that these mutations occur. And then, my guess is that some people understand that proteins are these things that go off and function and do work. But there's this kind of intermediate molecule that is really important in regulating a lot of things, and it's becoming more and more appreciated. And I'm sure RNA is become more familiar with the advent of like the COVID vaccinations. Um, so basically, what we study is this RNA has to be processed. Like when DNA becomes RNA, then RNA has to like have stuff happen to it
So
in order
normally,
for
like
it to then go in the.
it's too long. Like there's things we got to remove from it. We have to modify it a little
Yeah,
bit.
yeah. So we've got to edit this thing down so it can then go on and make proteins. And when this kind of intermediate like processing doesn't happen appropriately, we don't make good proteins. So that's what we're kind of studying. And kind of the brilliance of the cancer cell in this mutation is that you can have in other kind of mutations of cancer cells is you have one mutation which winds up affecting one protein, right? So you mutate the DNA and then that messes up the protein. But in our mutation, because you're mutating something that affects the processing of all of the RNAs, you can have mutation in one gene mess up a ton of different proteins. So it's a very elegant way for a cancer cell to kind of affect a ton of different things by only changing one thing upstream, whereas other mutations, you change one thing and then you get one downstream effect. So we're hoping that if we can kind of correct this intermediate processing step, that we can kind of treat these cancer cells without harming the healthy cells, which are doing this intermediate processing step appropriately.
Gotcha. So from chat, it's DNA to RNA to amino acids, and then the total protein structure, right? Would be like the step.
Yeah. So DNA kind of encodes like it's the instruction manual for absolutely everything. Like you want to make, a car, or do you want to make a cheese souffle? Like it's kind of all in the DNA. But certain types of cells don't need a cheese souffle. They just need, like a hammer. So you make RNA such that like you don't have to read all the DNA. You're just reading, cheese souffle. And then within that cheese souffle, there's a bunch of information which probably, is important for like making the cheese souffle, but like isn't important in the cheese souffle. So you cut out kind of all of that information that you don't need, such that you're only left with information that is cheese souffle. So that's kind of how we get from this DNA to this protein, is that like you take the section that you want and you take out all the things that you don't want in that section that you want, and then you're just left with what you want.
So we had someone come into chat saying I came in at the right time. We're sharing recipes.
I don't have a good cheese souffle recipe. I'm sorry.
So going with the cheese souffle analogy, the modification on the RNA, would that be something like using the right like plates to hold your cheese souffle? Like what are some of the modifications that go right or that would normally be like the good kind of modifications, and how do they go wrong, and what is the effect of that?
So you can imagine that like when you're reading a recipe, there's like a lot of like little bits and pieces that are like really important for you as like the recipe reader that aren't really important for like the recipe. So like, if you're reading a Julia Child's whatever, and she's like, oh, and I greatly prefer to use this elongated wooden spatula rather than, a short plastic silicone spatula. That's really important for you as the reader to like see that, but like you don't want the wooden spatula to wind up in the cheese souffle. So when the cell's kind of going from recipe to product, it's kind of like you reading where like the cell needs these bits of information in order to make the protein, but these bits of information shouldn't be included in the final protein. So we have to hack all of those out. And so you can imagine that if you're not appropriately removing kind of those user only bits of information, you wind up with the cheese souffle with the spatula left in it.
Gotcha.
Who wants melted spatula with their cheese souffle?
Well, I guess if it's mahogany, it's fine, right?
Oh, is that how that works?
So we have Lynn says, in undergrad, the research that I did was involved in studying how aberrant tRNAs in Saccharomyces cerevisiae are retroactively transported back to the nucleus to be fixed, and what actual mechanisms or enzyme assist in the specific pathway, and what would happen if the aberrant tRNAs are unable to return to the nucleus if fixed. So that's that's pretty cool. So it's always about how the RNAs are being modified and they're moving back and forth. Is there a particular RNA type that you're looking at, or is it just the effect of the modification itself?
So the mutation we study affects one of these things that takes out that like unimportant information. And so basically what happens is that like a huge portion of like messenger RNAs, so since we've we brought up tRNAs, we'll throw in a different type of RNA, which is our messenger RNAs. So these are the RNAs that encode the proteins. A ton of them get messed up. So we're just basically trying to figure out, like, which messed up RNAs may be causing problems. So can we target a specific messed up RNA um, to find treatments? And then the other question we're asking is, as a result of like kind of this global screw up within the process, like how is that kind of screwing other things up in the cell, and can we target those, because those are now messed up and not functioning appropriately? So are those a unique vulnerability to these cells, which, cells without this mutation, the healthy cells in the bone marrow, wouldn't necessarily be vulnerable to a little poke or a jab at? So it's kind of a double pronged attack. We're looking at specific RNAs themselves and asking, if I mess up this RNA, what happens? And then we're also kind of on the other end asking, since all these RNAs are messed up, what else is messed up as a result of that? And can we kind of poke at that to kind of cause these cells to die?
So is the end game to cause the bad cells to die or to correct them? Like, what is because you're talking about like drug development that you're working on. So like, what is your dream result in terms of the drug that you're working on?
So a dream result obviously would be to correct the error, but that's just not. Unfortunately, I don't think that is that's tractable. I think the as with any cancer treatment, right now the best thing that we can hope for is, can we kill all of the sick cells, and that will allow the healthy cells to come back and repopulate the bone marrow space.
Cool. So I guess, how does one go about making a drug like that? Like, when you started, like, did you already have a candidate? Did you have to do a drug screen? Like, and what is the phenotype that you're looking at? So when you're doing the experiment, like, how do you know that the drug is working? Do you have a mixed population of cells and you're killing off the bad cells but leaving the good ones? What is the day-to-day life of the soon-to-be Dr. Elizabeth look like when you're doing these experiments?
So in terms of the drug stuff, basically we start off with drug screens, where we take cells with or without our mutation of interest, and we just test drug after drug after drug, and we treat both the healthy cells and the sick cells with kind of a dilution series, so really really concentrated drug and really really dilute drug. And then we see, does this kill off, at all concentrations, does this kill off the cancer cells quicker than it kills off the healthy cells? And we actually have this wonderful and intrepid undergraduate student in the lab who normally sits right back there. And basically that's all he does, is he tests drug after drug after drug. And then when we find a group of drugs that's interesting, so maybe 5 or 6 drugs that all kind of hit the same cellular process, we then ask, okay, how? And then simultaneously, while we're asking the kind of the molecular questions, like how is this drug working in the cell to kill off the sick cells more than the healthy cells, we also do tests in our model organisms. So we also do tests in our mice to see, is this tolerable? Because not all drugs are tolerable in full systems, even though they seem to work in a petri dish.
Mm-hmm.
So that's kind of how we do kind of the drug stuff. And then on the flip side, kind of also what we do is we do sequencing. So we do big data, where we either take publicly available data, or we take patient samples and we sequence all of the RNAs in the healthy cells and in the patient cells. And we ask, okay, what's different? Who has more of a certain type of RNA? Who has more of another type of RNA? And can we link these groups of RNAs back to another cellular path? So we can kind of hit it from the front end, where we're just trying drug after drug after drug, and then we simultaneously kind of hit it from the back end and say, okay, and from what's changed in our cells, can we find a drug on that end as well? So two-pronged.
That's a really cool approach of having both directions, because not only do you find something, but then you work out the mechanism for how it's acting and try to as you're targeting normal cell, like not targeting the normal cells, but targeting the sick cells. We have some questions about that actually coming in. So handmade art is, I guess, how do you identify when a drug is working to begin with on the front end? So they also ask if you can use visual machine learning classifiers to see if the drug of interest is having the effect on the cell.
So how we know on the front end, super great question. How we know on the front end is that the cancer cells die faster. Like and hopefully what you would see is that like at a certain drug concentration, the healthy cells are still pretty alive and that the cancer cells are way dead.
So is it
And
just
like that right there is
like a dish that has normal cells and a second dish that has cancer cells, and you drop the same amount of concentration of drug on both, and then at certain time points you just go take a picture and you just count?
so we actually use a color, trying not to use all this snazzy lingo. We use a color changing assay, which allows us to, we have to kill the cells at the end point, but if there's a lot of live cells, it only changes color a little bit, and if there's a lot of dead cells, it changes a lot. And then we use this thing that reads 96 well plates. So basically it goes through and just like shoots a light through all the wells and then tells us the change in light from one well to the next. And so it's really, it's super simple. We don't have to go through and count anything.
Nice.
We throw some dyes on there and then have a machine take pictures, and then we plot the numbers next to each other. So it's really pretty straightforward. And then
so you could do
in terms of machine learning,
replicates and concentrations of one drug, like all of that on one plate. You could knock out like an entire drug per plate, if not more.
You could do 2 drugs per plate.
Nice.
If you plate them well and if you're good at what you do, yeah, you can. And the plate's like that big, so I think I've seen Evan with stacks of plates this high, and yeah, like you just rack them up and test them. So it's yeah,
that's amazing.
It's a really high throughput, good way to test drugs, kind of in this very synthetic environment.
Nice. And then sorry, then you were mentioning the machine learning element to it as well.
That's such a cool question, and there are entire labs dedicated to using machine learning and using kind of what we have now. I wish our lab worked on that, but it is far beyond the knowledge and expertise that we currently have. But there are lots of labs using machine learning models to kind of understand drugs and to understand cellular data to develop new drugs. So I think it's the wave of the future, and it's a great question.
Nice. We have some slightly caffeinated wondering, is there a specific molecular target that might narrow down the class of drugs you're testing?
That's a specific molecular target that might narrow. No. I think so for our drug screens, we're basically trying to use everything that's been FDA approved. So we're just like what's already being used in patients and what's already being approved. So we're just, I mean, if it's been published in the past by other groups to have worked in cancer X or cancer Y, but no, I think right now the goal of my boss is just like hoard all the FDA approved drugs and whack them on and see if they work. We've got the labor, so we might as well just give it a try. You never know what's going to work.
So is that then you can use it theoretically for off-target uses is the idea, right? Or like off-label, like it's already been tested for safety in humans, and then you can maybe fast-track it almost to treat a particular kind of cancer?
Absolutely. Yeah, no, there's been all sorts of amazing discoveries when we take, orphan drugs or drugs that are intended for one thing, and all of a sudden someone's like, hey, let's just see what it does. And, sometimes it works, so it can't hurt.
Nice. Handmade art, I was also wondering, what kind of data analysis techniques do you use on RNA sequences when you have such a large data set?
So I don't know how to answer that. So I personally do a lot of big data analysis. So wonderfully, the scientific community is all about sharing. Sharing is caring. So there are some really great packages that have been built, these bioinformatics packages that are, well published and well cited in GitHub. And the computational community here at Fred Hutch is absolutely amazing. So we have this brilliant infrastructure, we have this enormous cluster. So basically, I just kind of grab whatever package I want, and then I use Linux-based code, and I use R-based code, and I use Python-based code to implement these kind of packages on the supercomputer. And it allows me to determine what RNAs are being expressed by using reference samples, kind of like the whole human genome. And then once I have those, I can determine differential RNA using statistical packages, um, some of which were invented here at UW. And then from there, I can look at different pathways. So like what cellular pathways are being affected using other packages as well. So there's a world of scientific computing out there. And again, the scientific community is all about, let's get these tools into everybody's hands so that we can really understand what's going on.
I love there's a question in chat. Do you use DESeq too? The particular
I do.
There you go.
DESeq too.
See, this is I just why I also love the community. There's like such detailed questions that come up, even like the particular package that you're using. There's like someone out there who's like, yeah, I know that one.
Yeah, I was trying to avoid all those kind of little names because I didn't want to. But yeah, no, DESeq 2 is my jam.
Yeah, just Science Campus is just spicing it up. Just spicing it up with their questions. So with your drug, I guess how did you come to your drug of choice that when in the lab did you get to choose what drug you wanted to study, and then what is your mission with this drug that you're trying to ask?
So for the lab, we just published a paper. I think it just came out where we found a group of drugs, and we didn't get to choose it. We just found what worked. So like the drug kind of picks itself. And so that was just done by our wonderful postdoc. She just published on a group of drugs which we think are a novel vulnerability for our specific mutation in our specific cancer. And then for me, I actually am not working on it from the front end with the drug screens. I'm working on it from the back end. So I've picked a handful of targets. These are things that we've found to be misspliced using kind of the bioinformatics, so doing the big data stuff. And I didn't pick that either. So before you join a lab, you do what's called a rotation, where you just kind of like hang out in a lab and like, does this work for you? And my boss was like, hey, I see that you used to work with this protein. We found this specific target. Would you mind working on it for your rotation? And I'd published on this protein before, so I was like, yeah, sure. And here I am 4 years later, still studying the same target. So it kind of, again, I guess I didn't pick it, it kind of picked me. But there's always a reason and a rhyme to kind of why we pick what we pick, and that's usually because of pre-established data in the field. But then I've been kind of picking things on the side as well, of things that I think are interesting to kind of hit on the back end of potential pathways. So
what can you tell us? And of course, like if there's anything you can't tell us because it's not yet published, please don't ever feel pressure to tell us about it. But like in terms of the mechanism, what is it that you're studying? Like what is like the nitty gritty of what the drug is trying to do, and how are you studying that question?
So for the drug that we just published, in your cells is like I said, as things get old, they break. Like in your car, like you're gonna throw a wheel bearing, your transmission's gonna go out. So the same is kind of true for our cells. That like as a cell ages, the DNA can actually get a little wonky. It's kind of like reading and photocopying a book over and over again. Like you can imagine that as you read and photocopy a book, like everything just kind of wears and tears and falls apart. So as our cells age, that happens to our cells. And the DNA can break. And there's this pathway called the DNA damage response pathway, which is supposed to come in and fix everything. But, none of us are infallible. And so what we've recently found is that in our specific mutation, that this seems to be a unique vulnerability. Even though RNA is downstream of DNA, somehow when we mess up all the RNA, something weird happens with our cells' ability to fix their DNA. So we're trying to target this upstream error, which has been shown to be a unique vulnerability. And in our paper, we're showing that this seems to preferentially kill cancer cells in a dish, and also works in our mouse models, both against murine cancers and when we take patient cancer cells and put those into mice and then treat those cancer cells in the mice. So we're really hoping that this kind of paves the way for this new drug, this drug which is already being used in other cancers, to kind of find a new home with our specific mutation.
That's really cool.
And then for my stuff, kind of what we're thinking is, again, we're looking upstream. So DNA becomes RNA, RNA becomes protein. It turns out that there's a whole host of these RNAs that encode what helps the DNA make RNA. So like you use a protein to turn DNA into RNA. And that protein is encoded by an RNA. And with our mutation, the RNAs that make the protein that turns the DNA into RNA can also be messed up. I know that's a lot, and it's hard to even my grant reviewers have a hard time understanding it sometimes. But so there seems to be some more stuff that when you mess this up, this gets messed up. So I'm looking at these specific proteins that turn DNA into RNA, and how those are misexpressed, and hopefully we can hit this again in another way and kind of mess up this kind of core process within these cells.
Nice. You already kind of answered one of the questions that popped up, which was how much overlap is there between the different type of cancers? Essentially, if you're working on drugs that have mechanisms that are broadly applicable to multiple kinds of cancers. But you were mentioning, like, the drug that was just published, right, already had been approved in other cancers. And so the idea is that you'd be cross-targeting and could be utilized in other cancer studies too. Is that correct?
Yeah, so that's a really big question. There's some, depending, and that's about as like, it's, cancers are so, they're almost like people. They're all a little different, because everybody's body is different, and everybody's been exposed to different things, and everybody's DNA is different, even if we're getting mutations in the same things. So there's some depending.
Gotcha. And then there was an interesting point you made too about how you tested for this. So you made a tumor within a mouse. That was a mouse tumor. Then you mentioned that there was also the potential of grafting on a human tumor. Can you go into a little bit about how does one do that, and then how do you test the drug in that kind of model, like where it's like you're essentially humanizing a mouse, right?
Yes, so they're very specific types of mice that have been genetically manipulated such that they can accept human cells, and we are very fortunate in that, because our cancer is blood cancer, we can basically collect cancer cells by just collecting blood and then isolating the specific population of cells that we're looking for. And then once we have this core group of cancer cells that we've isolated from these patients with these diseases, we can then just inject them into the mice, and then those take up residence, and we can then study those kind of in a more native environment. So that's in an animal. And then for our drug studies, we generally just try to do conversions. So like, what do we treat people with? Has this been used in mice before? Can we just like scale things up and scale down? So then once the cells have kind of like taken hold inside the mouse, we just treat the mice on an experimental timeline determined by multiple factors and see, do the cancer cells persist? Does the mouse get sick? Because that's another question. If we kill the mouse trying to kill the cancer cell, the treatment obviously isn't going to be viable. So it's there's a lot of nuance that goes into it, but basically, yeah, we use a genetically modified mouse with cells that we've just collected from a patient's blood.
So you'd be able, in theory, to do the profiling on that patient if you wanted to, and then go and actually put it into the mouse. So you'd have some human answers already, and then maybe you can work on the human cells in a dish too, and then put it into the mouse. So you can go like a lot of the different levels, almost like from human back down, like down into the mouse and back. And Kite Fish, thank you for the $50 donation. We're at $13.70. Thank y'all very much, chat.
Wow. So we can hit it from all angles, and unfortunately, just because things work in a mouse doesn't mean that they work in a human, but it's a good step in the right direction.
How do you know when it's working? If it works in a mouse, and then what's the next, like, what would be the next step that you would do? Like, how close can you get to a human? Like, when do you know if it's going to work in a person or not?
Oh, that's a hard question. That is way beyond my pay grade. I know that you have to do a bunch of mouse studies and then something, and then you do your first round of clinical trials. But that something in the middle is that's way above what I get paid to know. So,
Gotcha.
Hopefully someday I will be able to answer that. But yeah, I think it's a lot of red tape. Is
Gotcha.
the something in the middle.
I wasn't sure if there's like some kind of protocol with like certain cell types from humans before you make that leap, that phase 1 clinical trial, because that always seems like the hard leap to do, right? Like you were saying, is how much evidence is enough evidence to say we're ready for that human trial, and like that everything works good enough to actually do that testing.
That's one of the reasons why we try to work with FDA approved drugs, is so that we're not coming up with anything new. We're just hoping to say, can we shift from one to another? And the brilliant part about working at the Fred Hutch is because we're also part of the Seattle Cancer Care Alliance and University of Washington. There are people on my floor who are physician scientists, and we can walk across campus and there are doctors. So the hope is that, and again, beyond my pay grade, but we can make the findings, especially since we're using FDA approved drugs, and then walk across and talk to doctors and hopefully find cohorts for clinical trials.
Nice. That's a really nice, I guess, benefit to working at the Hutch, is that you have that access to the doctors as well firsthand, so you can get that direct feedback from the clinicians, which is really exciting.
Yeah.
Handmade Art says it's more of a statement than a question, but it seems really wild that you can inject a mouse with human cancer cells and it'll take up residence there. Is there something you can give? That's something you can give an idea of how that works.
Yeah. So if we're talking about our bone marrow cells, like our bone marrow stem cells, they want to live in the bone marrow. Like that's their home. That's where they're happy. The surface of all of our cells are like decorated in all sorts of different proteins. So with our mouse models, I kind of skipped over a few steps with kind of how that works, but basically what we can do is we can irradiate the mice so that they don't have any of their own bone marrow cells. So there's plenty of space in the bone marrow. And then we inject these bone marrow cells from patients, or these cancer cells from patients, into the mice, and they want to live in the bone marrow. And since we've eliminated the mouse bone marrow, there's plenty of space for our cells to just kind of like, they wiggle in there and they make themselves happy.
And so is that an immune compromised mouse that you need because you're putting in human cells?
Yeah.
Okay.
Yeah, so there's a handful of them. So we basically they're altered such that they don't express certain things, and then once we eliminate all of their kind of blood cells, then we don't have to worry about them making new things that could come and attack the human cells. And then we've put some human genes in there as well, such that the mice kind of express some stuff that make the human cells happy as well. So they're really highly edited mice. But,
nice. We're not just taking like a street mouse or a common wild type mouse from like Black 56, but a very specialized mouse to run these very specialized experiments.
yeah, these are very well taken care of, very very expensive mice. They live in very 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 clean environments because we've messed around with them so much. They get sick very easy because we've already decreased their immune system, and then we're irradiating them. So we yeah, they're very very specialized mice.
Nice. That's really, really cool. And Ham, thank you for the $100 donation. Y'all, we are $30 away from running that first set of raffles there. So thank y'all very much for that. Because if we get to 1,500, we'll do a raffle. Maybe with Elizabeth still here, she'll get to tell us when to roll it. And that would be kind of cool. Dacum was wondering, in choosing the drugs to try, does anyone look for any correlations in different cancer cell structure, behaviors, any factors other than just random?
Oh yeah, I mean there are definitely super targeted ways of going about things. In my first lab, what we would kind of do is we'd kind of do the back end stuff, where we would find kind of all of the signatures. It was definitely kind of like a mechanism forward lab. And then we would find, these cellular processes that were messed up in that specific cancer type. And then we would kind of look for all of the drugs that possibly hit a bunch of those different cellular processes and then test them kind of like that. So we do kind of a back end forward kind of methodology. So you can absolutely do it both ways. The reason why we're kind of testing everything that we possibly can is because so blood cancer is very very well studied, and so there's been a lot published but not necessarily a lot of headway. So kind of the idea is that if we kind of cast a wider net and we look really big and really broad, can we loop in more things? And it's not totally random. Like we definitely there's some thought to kind of what we're grabbing, but I think the idea is the wider net that you cast, the more likely it is you'll kind of reel something.
Why do you suppose, and maybe this is this is a kind of a difficult question, but why is there so much published but so little headway?
So for our specific cancers, I think it's super complicated, and then we're also limited to accessibility of things like patient samples. So for things like AML, which are super super common, it's very easy to get patient samples. But with MDS, there are far fewer samples, and because they're age related, I think the kind of the key is doing longitudinal. So can we catch people before they get sick, like well before they get sick, to kind of find out what happens that kind of gives rise. So that's another limitation. And then for our specific mutational group that we're looking for, a lot has been tried and just a lot hasn't worked.
Okay.
So there have been a bunch of different clinical trials trying to hit splicing specifically, and it just they had poor outcomes. So it's very complicated. So I think it's multi-pronged and multifaceted. The more you kind of zoom into our specific
Is
our
the
specific field.
is the possibility of using like these pre-approved drugs? Do you find it makes it any easier to like the likelihood to make it successful?
We're a newer lab, so maybe, hopefully.
Maybe, but no guarantee
Yeah,
on
we've
that either.
no. I know, and again, just because something works in the petri dish and something works in the mice doesn't necessarily mean that it's going to work in people. Because we've obviously had, like as I mentioned just a second ago, there were clinical trials for drugs targeting the specific mutation, that looked amazing in the petri dish and looked amazing in the mice, and then it went into people and it had some scary side effects, and so it was all canned. So. Um, we hope that using FDA approved drugs means that we can find something, but there's no guarantees, unfortunately.
Yeah, again, I think that highlights the complexity of the question that you're studying, and how it's not at all easy. And the heroic efforts done at the Hutch by researchers like Elizabeth is what hopefully will beat cancer one day. So thank you for everything you do. I know it very much feels like it's a day-to-day basis. You're just pipetting away, and I'm sure it's kind of hard to get. I know for me, it was really hard to get out of that mindset that you're doing for working on something really bigger than what's just in the lab, but there's just so much that you can be doing, like that you're doing, that furthering the knowledge to hopefully help people one day, which I think is, again, really, really amazing. We had another question from Ham. Is there a holy grail that you are looking for? Like, what kind of hypothesis are you testing, or is it more just looking for what works and what doesn't?
I mean, the holy grail would be we find like the one cellular process or the one drug that that just works, right? Like it eliminates the cancer cells and it keeps all the healthy cells healthy, and you give it to the patient for a short period of time and it's already FDA approved and it's cheap. Like that's the holy grail, right? Like you find something that like really helps people really fast, and then we can get into the clinic really well. That's kind of, I think, on the cancer treatment side. And then on the flip side, kind of the holy grail hypothesis to prove would be like, oh, this is the one thing that these mutations are doing that leads to the development of this cancer. So it's kind of two pronged. Like, can we understand the basic biology? Like, is there that one thing where we're like, aha, this is why this causes cancer? And then on the flip side, can we treat that? Alas, we have not found that yet. Someday.
I have another naive question for you. When you're targeting these cancer cells, do you by chance know what mechanism allows the drug of interest to target the cancer cells preferentially, or is it just more of like that color reader that you're looking at of like targeting some cells versus another? Like how does one know it's like a good candidate for that step, right, of like targeting the bad cells versus not the bad cell? Like what is the connection there?
So with any mutation, it's kind of like, I don't remember the name. Those giant machines where like you knock a thing, like the Pee Wee Herman used to have that made him breakfast. So like that's kind of the way our cells work, is that like everything kind of leads into everything else, and then unlike Pee Wee Herman's breakfast machine, it does more than just makes breakfast. Like it makes breakfast and like also gets the clothes right. Like there's like a ton of different things, so it's a cascade from like one thing down. So the way we would kind of pick, can we target these as a vulnerability in cancer, is based upon, every scientist stands on the shoulders of generations of scientists before them. So we can go back through and look at the literature and say, okay, well, we know that this has been shown to be involved in this, and then maybe there's a hint of it being involved in this, and like it's really well established that it's involved in this. So we can kind of pick things that we know are kind of in the realm of what's messed up when you just screw up this one thing here. So there, by using kind of the work of so many other great scientists before us, we can kind of hone in on things which may be a specific vulnerability for our cancer cells of interest.
Nice. So it's there's a lot of back end work that you're using to build the current hypotheses to drive you forward, and that's what's kind of allowing us to happen like that. And Ham, no sorries for questions. Please ask your questions. Is there a particular cancer that has been solved and sets a template for how to approach this kind of research?
So is the question, is there a particular cancer that has been solved?
So like maybe almost a roadmap of how you can answer these questions in other cancers. Like, is there a cancer type that we figured out, we've cured, we know exactly how to do it, and then you can just base all subsequent approaches based on that one approach?
No, there isn't. We have no one cancer. We've gotten really good with some cancers. We've gotten really good, and like we've really like made sure that like your chance of survival with a certain cancer is like really, really, really good, but we have not cracked the nut on any of them, I don't think. And I could be wrong. I have not read all of the literature, but as far as I understand, like, again, cancers are like people. Like every cancer is different. So you could have that one cancer where 91% of people survive, and it almost always responds to this type of treatment, and you are that one unlucky person that for some reason it just doesn't. So I wish there was a roadmap. It would make my job easier, probably a little more boring. But yeah, there's unfortunately, I think that for all researchers, we all kind of, there's a methodology to how we approach things. And I think the majority of us kind of follow that, either come at it from the front end with the drug or the back end with the molecular mechanism. So I think that's the closest thing I would say we have, is that like we have ways of asking questions, these defined kind of methodologies for how we go about systematically approaching these questions. But outside of that, sorry.
No, that's a great answer. Thank you for that. So actually, I want to be respectful of your time. We've got a couple of minutes left with you. We got another $40+ donation, so we're over 1,500. Thank y'all very much for that. So any last questions for Elizabeth, chat? Make sure to get them in now. I want to be respectful of her time. She's got to get home. I think she might even have a few last experiments to be able to finish out. So like y'all, let's not keep her too long. If you have any last minute questions, or Elizabeth, if there's anything you want to leave us with in terms of just reflecting or just top of mind kind of thing.
I mean, I guess I'd like to say thank you to SciAnts, and thank you to everybody who came. It was absolutely my pleasure to share kind of what I love with everyone else. So that's all I have to say.
Well, thank you very, very much for your time, for prepping these slides and walking us through. Like, it's just crazy to me how cool the nuclei look for the 6 cells versus the normal cells. And y'all, we're gonna look through these slides even more, but while Elizabeth was here, I wanted to make sure we got our questions in. But I thought it would be cool after Guppy comes on too, to look at some of these cells and see all the different kinds of shapes and nuclei that we can get, because it's madness, right? Like these cells down below by Elizabeth, like how wild they look and how different they are, and that's the disease state versus the normal state. And it's just really cool that you can have that kind of visual cue, and I'm seeing this under the microscope. I think really makes a world of a difference to really inform us of the importance of what it is that you're doing. And just thank you for taking the time, walking us through the experiments, your work day, your background. It's your inspirational story about getting into science, like, from the shot up car to now curing cancer. You are incredible.
Oh, well, shucks, thanks, mister. No, I greatly appreciate that. And yeah, I guess I'm going to go take care of all this stuff sitting on my bench that I probably should have taken care of before I got on here. This is my turn to ask a stupid question. Do I just hang up?
You just hang up. Thank you so much, Elizabeth.
No problem. Bye, guys. Thank you so much.
All right, y'all. That was Elizabeth coming on the stream with us.